superphylum ecdysozoa. These are your protostomes that have a cuticle or exoskeleton that they will periodically molt or shed in order to allow them to grow. So they are known as the molting protostome because they go through a process called ecdysis. So these are the molting animals or the molting protostomes. Two big phyla and two small phyla of ecdysozoa. So one of our first big phyla in terms of the number of species is the phylum Nematoda. Some folks say nematodes, like SpongeBob. Some folks say nematodes. Most of the folks I know who study them call themselves nematologists, so I'm going to go with nematodes. Diverse. They are an extremely diverse group of animals in terms of their habitat. They are found just about everywhere, and there are lots and lots and lots of individuals out there. Many of them are parasites on animals and plants, but there are also a number of free-living nematodes as well. They only have longitudinal muscles, which means they only kind of squiggle a bit. And I'll show you a video of that in just a minute here. Morphologically, they're fairly simple. So they all kind of look more or less the same, even though their habitats and their modes of transmission vary widely. If you want to find out more about nematodes, I'm just going to warn you, there are a lot of disturbing images out there on the internets and out in the world of nematodes and the damage they can do to animals and plants. Well, the plant ones aren't as gory, though. So I've got a list here of examples of nematodes. And apart from vinegar eels that you saw in lab, Turbotraex aceti, the rest of these are all pathogenic or parasitic. So I'm going to go and tell you about each of them in a little bit of detail. But the most famous nematode of them all has got to be this one, C. elegans. The C is short for the generic name, which is Caenorhabditis. Caenorhabditis. But even people who study Caenorhabditis elegans call it C. elegans for short. And speaking of short, you can see these are small little worms, only about 1 millimeter long, which means you can raise a lot of them in a very small space, which is one reason why they're famous, because they're a great model organism for researchers. And they're most famous for their developmental biology. As you can see, this particular nematode, not super sophisticated in terms of the number of types of cells and tissues and organs that it has, so it makes a good, simple model for developmental biology. Once it goes from being a zygote to dividing into 2, 4, 8 cells, researchers have been able to map the fates of each of those cells. And from the time it's a zygote till the time it turns into one of these adult worms, biologists have been able to resolve what's going to happen to cells through time by tinkering with their development. And this is one of the first animals to have its genome, the DNA instructions for making a C. elegans worm, completely mapped. So the genome has been resolved for this organism for a relatively long time in terms of genomics. So one of our other examples is Turbotraex aceti. This is the one that you saw in the lab. In case you don't remember, my next video on this playlist is going to be some images of them squiggling around. They're called vinegar eels. Vinegar, I think I misspelled that. Oops. They're not truly eels, but they do live in vinegar, and they feed on yeast and bacteria that are actually what cause beverages to ferment and become vinegar. Vinegar, the word vinegar comes to us from the French vinaigre, which means sour wine. All right, so let's have a look at some of these vinegar eels before we get grossed out by some parasites. 1,